Hi all, this is Bhavani, lecturer from Department of Physiology, SRM Dental College, Ramapuram. So today we are going to see about WBC. So WBC is white blood cells. The other name is leukocyte. So this is an important cell which is present in our blood. So this blood cell has been formed by the process which we call it as leukopoiesis. So the process of development and maturation of a white blood cell, that is your leukocyte, is called as leukopoiesis. So this is the flow chart to show how a granulocyte has been formed. So the starting is from your hemocytoblast. From that, the myeloid stem cell has been formed. From there, myeloblast. From myeloblast, it is converted into promyelocyte. Next, it has been converted as myelocyte. From myelocyte, it is metamyelocyte. And from there, the band formation has been done and it has been differentiated into granulocyte. So this process has been done through a myeloid series. So this process we call it as your granulopoiesis. Through this process, the granulocytes are formed. So next is a formation for a monocytes. Monocytes, they play a vital role. So the monocytes, when they are present inside the blood cells, we call it as monocyte. When they enter into the tissue level, we call it them as your macrophage. So these cells are being formed from hemocytoblast, from their myeloid stem cells, from their monoblast has been formed. From there, the promonocyte has been formed. From there, the monocyte, which when it is present in blood, we call them as your monocyte. So when they have entered into a tissue, we call them as your macrophage. So this is the formation of a monocyte. So next is lymphocytes. So this is process, we call it as your lymphopoiesis. The hemocytoblast is the starting cell. From there, lymphoid stem cell has been formed. From there, lymphoblast has been formed. From there, lymphocyte has been formed. From there, the lymphocyte which enter into a plasma cell. So these lymphocytes, they play a vital role. So next is regulation of leukopoiesis. So the granulopenia or dead granulocytes and monocytes, these persons are released by GCSF, then MCSF and GMCSF, then interleukins. So based on this, they stimulate bone marrow. So from bone marrow, the formation of granulocytes and monocytes are formed. So the normal counts, when it is in a normal count, it goes and inhibits your bone marrow so that the increased formation of the cells will be formed. So this is a regulation of leukopoiesis. So when the WBC cells has been reduced, the bone marrow will be stimulated so that they produce your monocytes. When they have increased, the inhibitors will be created so that they stop the production of your granulocytes. So the normal range of WBC at birth, in full term, the infant will be having 10,000 to 25,000 ml of blood. In infants up to 1 year of age, it is 6,000 to 16,000 ml of blood, and for an adults, it is 4,000 to 11,000 ml of blood. So here you don't have any difference between male and female because immunity is common for both. So you doesn't have any difference between male and female like RBCs. So next, the variations in your WBC count. So when the count has been increased state, that is more than 11,000, we call them as leukocytosis. So the physiological changes and pathological changes are, in physiological changes, the age, exercise, mental stress, pregnancy, after food take, then experiencing to a low temperature will increase your WBC count. The pathological conditions are acute bacterial infections, the spirogenic organism formation, then burns, post-operative period, tuberculosis, then granular fever. During these situations, the WBC count will be more in number. Next, when the count has been reduced less than 4,000, we call them as leukopenia. Infections by non-pylogenic organism, example typhoid fever, viral infections, influenza, smallpox, mumps, etc. Then protozoal infections, starvation and malnutrition, aphasia of bone marrow depressions. During these conditions, the WBC count will be in a reduced state. Next, classification of WBC. So we divide your WBC into two types, granulocytes and agranulocytes. So granulocytes, we call it as neutrophil, basophil and eosinophil. and A granulocytes are lymphocytes and monocytes. So why we call them as granulocytes? Because they contains granules in them, so we call them as granulocytes. Why we call A granulocytes? Because we can't find any granules in them, so we call them as your A granulocytes. These 5 types of cells are being classified based on their nucleus which is present inside them. So based on the nucleus we differentiate them into neutrophil, eosinophil, basophil, lymphocyte and monocyte. So the neutrophil will be having multi-lobed nucleus. The eosinophil, that is more than 2 lobes, we call them as your neutrophils. Next, eosinophils and basophils, they both are bilobed structures. These two cells we can find only during the infectious condition. We can find these 2 types of cells. Lymphocyte is a large spherical structure. So lymphocyte is, again, it has been classified into 2 types, small lymphocytes and large lymphocytes. Next is monocyte. This contains a bean shaped or kidney shaped nucleus in them. So monocytes also play a vital role. So neutrophils is a major cell which is plays a major role during your immunity process. So thank you.